let's go over today's homework first. So the first thing you need to look at is, all right, so uh, number one, in the P generation, the male guinea pig has black, rough fur. These traits are both dominant. So he has capital B for black, and capital R for rough. All right, the male guinea pig is homozygous for both. Again, it says it was a dominant trait. It also tells you that it's the P generation. The recessive traits are brown soft fur, which is basically recessive. And in order to be recessive, you got to have recessive for both traits. So it should be lowercase b, lowercase r. So if you FOIL this, the first B goes with the first R, the outside B with the outside R, the inside B with the inside R, and the last B with the last R. And so all of those are going to be capital B, capital R for all four squares, which is right there. All right, the female, lowercase b, lowercase r, again, FOIL it. First B, first R, outside B, outside R, inside B, inside R, and last B, last R. And those are all lowercase b, lowercase r. The F1 genotypes would be all capital B, capital R, lowercase r, and the phenotypes would be black and rough fur for all of them. You didn't have to answer those two, but yeah, that's what the answer would be. All right, in cattle, a brown coat is dominant over red, and hornless is dominant over horned. A horned red bull, so horned tells you it's recessive, so lowercase h, and red is also recessive, so lowercase b, is crossed with a female that's heterozygous for both traits. So when you FOIL it for the male, it's going to be lowercase b, lowercase h for all four squares. The female, all right, so first B, first H, so capital B, capital H. Outside B, outside H, capital B, lowercase h. Inside B, inside H, lowercase b, capital H. And then last B, last H, lowercase b, lowercase h. When we look at here, green color is dominant over yellow, and smooth is dominant over wrinkled. Cross 2P plants heterozygous for both traits. So this is just like the problem that I went through in class in the last video. And so again, you're going to be crossing capital G, lowercase g, capital S, lowercase s, and you FOIL that out, and you get this and this, and then, all right, so green smooth, green smooth, green smooth, green smooth, green smooth. This one is green wrinkled, green smooth. This one's green wrinkled, green smooth, green smooth. This one is yellow smooth. This one is also yellow smooth. Green smooth. This is green wrinkled. This is yellow smooth. And here we have our yellow wrinkled. So that is correct. It's a 9-3 to 3-1 ratio. All right, come down here. In a dihybrid cross, when two traits are considered, the number of possible combinations in the offspring increases. Suppose that you have black hair, capital B is dominant over blonde hair, and that brown eyes are dominant over blue eyes. What percentage of offspring would be expected to have blonde hair and blue eyes? So this is the father's genotype, capital B, lowercase b, capital E, lowercase e, and the mother is blonde hair and blue eyes. And so there we go for each of those. And so this is correct, and notice that the blonde hair, blue eyes are all in this column. That is 4 out of 16, or 25%. Now, both parents have black hair, heterozygous, and brown eyes, heterozygous. So you put that there. This is just like the one up above. It's a 9-3 to 3-1 ratio. Up here, notice it said black hair. Down here, it says brown hair. It's the same difference. It is a typo in the worksheet. But anyways, it's 1 out of 16 have blonde hair and blue eyes, which is 6.25%. Okay? All right. So anyways, that was yesterday's worksheet. If you have any questions, just let me know. And today, all right, we're continuing with this, but we're basically interpreting the results just a slight bit differently. All right, so when we looked at these Punnett squares that were 16 by 16, we had a 9-3 to 3-1 ratio. Let me see if I can find one here. Here we go. Okay, so it's a 9-3 to 3-1 ratio. Okay. And that's pretty straightforward. But what we found is that sometimes we have a 9 to 3 to 4 ratio. And so we had to figure out what the heck was going on with this 9 to 3 to 4 ratio. And this is what we call epistasis. Epi means on top of, and stasis means state. So it's on top of a state. In other words, this is a state that blocks the expression of another trait. So for example, you have black labs, you have chocolate labs, and you have yellow labs. And one might just expect, well, it's a incomplete dominant trait. Well, if that's the case, you'd have a 1 to 2 to 1 ratio of black to chocolate to yellow labs. But what we end up having is a 9 to 3 to 4 ratio where the chocolate is the 3 and the yellow labs are the 4. Also called golden labs sometimes too. You have your golden labs and your Labrador retrievers. Yes, they're different breeds, but the color patterning is the same. So the color depends on the allelic combination of two independently assorting genes. And so basically the second gene blocks the first gene. Basically, it's just complete dominance where black is dominant over chocolate. Well, what about yellow labs? That is where the second trait comes in. So basically, first gene, capital B is black, lowercase b is brown or chocolate. Where the second gene comes into play later. So just hold on a second before we go there any further. So if you have capital B, it would be black. Capital B, lowercase b, again, black. But if it's lowercase b, it's brown. But there's a second set of genes. And if it's a capital E at all, if it's capital E, or capital E, lowercase e, nothing happens. The epistatic gene is dominant in terms of if you have it, there's no change. But the epistatic gene is recessive when you have two copies of it. Now suddenly it blocks the production of this first one. So two recessive lowercase e's blocks any production. So even if you had capital B, that no longer matters because this lowercase e is a different gene on a different chromosome that then blocks this gene from being expressed, and now we have a yellow lab. And here you can see the different phenotypes. Capital B at all is going to be black as long as on the second one we have a capital E for the epistatic gene. Here we have lowercase b's and they're going to be chocolate as long as the second set of genes has at least one capital E. And down here we see with a lowercase e, that blocks whatever we have here, and now it's just going to be a yellow lab. Okay, so here we have another example, and actually one of your problems does this, except for it switches the epistatic gene and puts it first, I believe. So you do have to be careful with that. It's like black and brown mice. The brown is also known as a goody. And then it's like two lowercase a's are going to be albino. Here they use lowercase c's as the second gene, and again that's albino. It blocks whatever it is. So the dark gray or black is dominant over the brown mice, except for in the case where you have these two lowercase c's on this problem, on your homework problem, it's going to use, I believe it's a's, two lowercase a's are going to block it because that's albino. Same difference, white, albino. We also see this in horses as well. People who are into raising horses always correct me on the colors. I believe this is chestnut and this is bay. I like to call it dark brown and light brown. I do know this is called palomino. And palominos you only get if you have two recessive alleles and it blocks the production of the protein of the first one. So that's basically it. I mean, it's the same type of Punnett square that we saw just the other day, except for it's just the way you read the results. The second set of letters blocks the first set of letters. Another thing you can do with these Punnett squares is you can have incomplete and codominance. And so when you do that, you're going to have a wide variety of results. So let's just say it was like B's and W's. If we had a BB, that might be like black. BW might be silver. So like if this were a BW and this were a BW, that would be silver. And lowercase b might be like white. And so, that's for the first trait. And let's say the second trait was, instead of C's, let's say it was like a ring size, and so you can have capital R or lowercase r. Capital R is a big ring. Capital R, lowercase r is like a medium ring, and lowercase r is a small ring. And so, this would, you would have differences there. Basically, what's going to end up happening is you're going to end up with a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio when you have those incomplete and codominant traits. I am going to have you attempt at least one of those today. So, again, those problems are, you just got to think about it a little bit more. All right, so here we go. Typical dihybrid 9:3:3:1 ratio. Remember with the epistatic where it's double recessive, here are our recessive, so then it becomes 9 to 3 to 4 ratio. And I showed you this the other day. Remember, these Punnett squares can be absolutely gigantic when you start looking at 4 or more traits. Another thing that kind of happens is called penetrance. It's a ratio of how many persons with the phenotype have the genotype. So, for example, with the breast cancer gene, there are actually two of them, but if a woman has the BRCA1 gene, there's an 85% chance of getting breast cancer. In other words, it basically is a very highly penetrant gene. In other words, if you have this, you have a really good chance of getting breast cancer. It doesn't basically create the breast cancer. What it does is it's a gene that malfunctions, and it's basically you got to have like 7 or so mutations in order to get to where you have a cancer. And this particular one basically just makes you more susceptible because you already have one mutation out of the way. And so this is why it just basically really increases your chance. Notice that not everyone that has that gene, though, does develop breast cancer. And there's a variety of reasons for that. Unfortunately, some people pass away before they would have even developed it. Other times, the body just is, you have a really good immune system at finding those cancer cells and getting rid of them. There's really good environmental circumstances that impact that. So that's what penetrance is. Expressivity is a disorder can be expressed more or less severity in individuals. A person may have the gene for basically hypercholesterolemia, which is hypercholesterol, high cholesterol, but can have a mild or severe form of the disease. In other words, you have the genes to have high cholesterol. And maybe in some people, they have that gene and they're battling high cholesterol their whole life. Other people might have that same exact genetic code, but for whatever reason, the total makeup of the body is that, yeah, they have high cholesterol, maybe a little bit, but not, terrible to where it's life-threatening necessarily. They just kind of gotta watch what they eat a little bit. Other people, they watch what they eat, and it doesn't make a dent in their high cholesterol levels, and they have to be on cholesterol medication. Another example I like to think of for expressivity is, for example, a person can have the baldness gene, let's say a male does, and he has it and he is completely bald, no hair on his head at all. Whereas another person might have the same exact genetic code for being bald and they have like a mild to moderate receding hairline. Again, they both have the genes to have baldness, but we see a range of expression in that level of baldness. So, again, that's expressivity, another way that we see variation within human populations for the different traits. So multifactorial inheritance basically is what we've been talking about, where you have a combination of genes that come together to basically produce the outcome. Diabetes or cancer is a multifactorial genetic disease. In other words, like I said, a lot of times you have like around 7 or 8 or so mutations in a cell before it actually develops cancer. And sometimes you have genes that are already mutated at basically when you're born. So it makes you a couple steps closer. But like I said, a lot of times it's how the whole body works together. And then also just basically what's going on in the person's environment that can make differences. It's one of the reasons that we see subtle differences even in identical twins, because they have the same exact genetic code, but the way that those genes are expressed in the environment and just slight differences throughout their lifetime and their lifestyle, what they eat and such, can make differences accumulate. And actually, as twins age, they begin to look a little bit less and less alike over time. Okay, so here we have the worksheet I want you to work on today. I'm not going to have you do number 1 and 2. Number 1's actually really easy. You just had to FOIL. Number 2 is incomplete codominance, but students get a little confused with that one, and I'm just going to skip over that one for now. All right, so number 3 is one I was talking about with the epistatic gene. It's actually the epistatic gene, let's see, AA or capital A, lowercase a mice are agouti. All right, mice with lowercase a are albino because it blocks all pigment production. So yeah, it's the first one. All right, so either of these are brown, and if you have lowercase a, it's going to be albino. The second gene is going to be agouti or black. So lowercase b would be a black mouse. So just kind of a hint there. But the epistatic gene actually goes first in this one. Number 4 is an incomplete dominance one. I kind of want you to do that just because it kind of goes back to what we'd been talking about previously. All right, number 5, I'm going to tell you what the answer is before you even begin. It is a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. So it is a little bit more complex problem because you basically are going to have either big rings, medium rings, or small or narrow rings, and then they're either going to wash all their food, some of their food, or none of their food. This last one, you can refer back to the notes, and it is going to be basically black, chocolate, or yellow lab. So it's just work out that problem. These ones on the bottom, I am just going to skip for now. Basically, you should be able to do each of these extremely easily at this point. They're just review. They even tell you the type of question that they are, but you don't have to worry about those for today. Okay? All right. Thank you. This pretty much wraps up this unit, and good job. And next week we will be moving on.